And hello there. All right, I'm recording this on a Sunday afternoon. Hope you had a good weekend. Just before spring break in 2019, and this is going to be the DNA lab. All right, if you are a fan of Mr. B's Science Channel, you know that about 2 years ago, we did this exact same lab as part of our end of the year project that we did with the Dexter National Fish Hatchery, which is now called the Southwest Center for Aquatic Wildlife, I believe is what it is. I know that's close to it. But we just did this lab here at Hobbs High School last week, and there are several students that have missed it. So I'm going to perform that exact same lab as we did it in class. And for those of you that are watching from around, I'm going to describe to you everything that you need, and I'm going to let you know that I have a newer version of this lab paper that is going to be available. I've had several people email and ask me in the messages if some of my labs were available, and I go back through my files and most of them are not because they're just created for that particular time. However, this one, I will have a copy, an electronic copy of this lab in Word. If you would like to email me and ask for a copy, I'll be happy to send it to you. All right, for this lab, what do you need? Well, we have some strawberries. These are some that, as soon as I'm done with recording this, this is going to get tossed because they're about ready to go bad. You're going to need some distilled water. Dishwashing soap. This works the best simply because it has some added enzymes and proteins that help to break down organic material. You will need isopropyl alcohol, the good stuff. 91% is what we have here. And this we keep in our refrigerator in the storeroom. If you want to keep this in your refrigerator overnight, having it ice cold really helps. Now, different versions of this lab will ask you to use meat tenderizer. You can also use a pinch of salt, and I will show you how that works in just a moment. I also have some some stir sticks, just like you get in the famous coffee place. Now I'm using some scientific tools for this. I have a 10 milliliter graduated cylinder. This will show you how I'm making my solution that I'm going to smash with the strawberry. I also have a 100 milliliter one because I'm going to fill this half full with isopropyl alcohol, and then I have a 250 milliliter beaker that I'll be putting all of the substances in. The other miscellaneous things you'll need is a snack bag, a rubber band, and some cheesecloth. And I would say if you don't have a cheesecloth handy, if you have an old rag lying around the house, that might work. However, that might be too thick and it might keep all the DNA material from passing through. But you could try that if Mom doesn't mind. And then also you notice all of these materials you can use at home. All right, so what are we trying to do? We are trying to extract DNA material from some living organism. In this case, it's going to be a strawberry, and we're going to get a fairly large strawberry right here. First thing I'm going to do to the poor strawberry is I'm going to remove its hair. Set that aside. Now, we learned in class the other day that the most common variety of strawberry that's eaten in the United States is one of those that has 14 chromosomes, but it is octoploid. In other words, there are 8 DNA donors that go into making this. And you can look that up. It's still a little out there for my brain to wrap around, but it's a wild concept to look at. All right, we're going to put our strawberry into our snack bag. And normally I would have a bunch of this made up, but I'm doing this from scratch just so you see what the process is. We're going to set that aside. Okay, into our 10 milliliter graduated cylinder, I am going to be putting 2 milliliters of dish soap. And And we're going to fill that up to the 2 milliliter mark. If you're a little above or a little below, that is fine, but about 2 milliliters is what we want. I'm then going to add a pinch. Oop, that's a little too much. Let's see here. Yeah, okay. I'm going to add in a pinch of meat tenderizer. I know, it's a little more than a pinch. I don't know, what's the technical term, culinary arts people, for more than a pinch but less than a spoonful. And then I'm going to add water to the 10 milliliter mark, a little more than that, that's okay. More is not going to hurt us. You can see that I have the exact measurements written down. However, you can get by with a little more or a little less than this. And I'm just going to gently rock it back and forth and do a little mixing here so that we get the laundry detergent incorporated into our solution here along with the meat tenderizer. You can see I'm using that bubble to do that mixing for me. Alright, now I'm ready. Okay, DNA, as you've learned in biology class and science class, DNA is the blueprint for all life of living things here on Earth. That DNA in a human being is a double-stranded structure. Don't want to call it a molecule. It's a structure made up of millions of different smaller structures and molecules. But one DNA strand in our human cells, when it's stretched out from its folded state, can be over 1 meter long, 40 inches, more than that. What we're going to try and do is we're going to try and get the DNA out of this strawberry, because when we do that, it is much bigger than the cell that it's packed in. And because in this strawberry there's probably several hundred billion cells. You know that in humans, and it depends on where you look it up, human beings have like 30 trillion cells. Probably here in this strawberry there's several hundred billion cells. We're going to add our solution, which I just did. Let me see if I can get a few more drops out. I'm going to seal the snack bag. Now, I just want to show you something. I did not push the air out, and I didn't push the air out on purpose. I found that if I don't push the air out, I'm not going to burst the bag as easily for some reason. Then I'm going to take my fingers and I'm going to crush this strawberry. Now, a couple of things happening here. The physical act of me crushing the strawberry is destroying the cell structure. I am physically breaking billions of cells in this poor strawberry. The soap, water, and meat tenderizer solution contains proteins and enzymes through the soap and the meat tenderizer that are going to help the DNA be released from those destroyed cells. Now, you can also use iodized salt in place of the meat tenderizer. That's what we used when we did the previous DNA lab using the banana and the strawberry. I found with the meat tenderizer I had excellent results this last week. We're going to see if we get excellent results this time. And so I'm going to go around until the majority of the large structure of the strawberry is completely crushed. And as you can see, I've got some foam but not too much, but I have quite a bit of liquid in here from the strawberry. I'm now going to take my cheesecloth using my rubber band. I'm going to put the cheesecloth over the mouth of my larger beaker, and I'm going to prep here because I'm going to get my 50 milliliters, yes, 50 milliliters of isopropyl alcohol so I can float that DNA out. All right, 10 milliliters, 2 milliliters soap, pinch of salt and or meat tenderizer, fill to the max. You can fill it to the 10. I went all the way up to the top with distilled water. Snack bag, crush, create this. We're now going to take this and I'm going to pour this into my cheesecloth. And of course I had many students did not know where the name cheesecloth came from for this material, so explained where cheesecloth came from. If you don't know where cheesecloth came from, you can look at my previous video or simply Google it and you too will know why cheesecloth is called cheesecloth. Okay, using the stir stick, we can push down and see if we can get any more liquid through the cheesecloth from our pulp that's left over. When done, I can take the rubber band, take it off while holding on to the pulp here. I can take the cheesecloth and move it around, forming a holding structure. I can get a little bit more liquid out, and there we go. Put that in there. Now, does not look like I have a lot of liquid here, but in this liquid, I'm betting you, and I told this to the class, and I don't know for sure, but I'm betting we've got somewhere around 20 to 50 million DNA strands in here that we've liberated from the cells that we've crushed. We didn't get them all out, but we've got probably 15 to 20 million. And we can't see one with our naked eye, but you get 20 million together, and we can see those with our naked eye. Alright, so now here's the trick. We're going to hold this at an angle. Let's see if I can do it this way. Yep, do it this way. I'm going to pour the isopropyl alcohol down the back side. Now the DNA will not dissolve into the isopropyl alcohol. All of the solids will go to the bottom. The DNA will remain here in the solution. And I don't know if you can see this, but already there are strands of DNA coming together out of the solution, just like we did before. We're going to add all the rest of this in, and we're done. And here we go. Inside of here, I have DNA. Yeah, there's some little gas bubbles that are trapped, but all of this material here, there we go, that is DNA straight out of the strawberry. Millions and millions of strands of DNA. Now, students asked last week, what if we put our DNA under a microscope? Well, it looked just like mucus under a microscope. You wouldn't see any structures. We don't have a microscope powerful enough in order to see it. Alright, so what do we do with this information? We have some questions here. Question number 1 wanted to know, describe how a large DNA molecule is enclosed compactly within the nucleus of a cell. Well, remember that we've learned that it's folded and coiled inside the nucleus. Coiled, ED. Also remember, in eukaryotic organisms, DNA can also be found in the mitochondria. In prokaryotic organisms, the DNA is usually found in the cytoplasm. Describe the procedure from extracting the DNA. Well, first thing we did was we crushed the berry using a soap mix. We then strained the liquid. And in third, we observed the DNA. Describe the appearance of the DNA. Well, here's where we had some confusion. Students wanted to call that white down there. They wanted to call this white, when actually it's more colorless, although one student in my 4th hour actually used the word translucent, which is an even better word. So you could say whitish, translucent material. Explain the purpose of each one of these. Now this, I've taken this out of the new lab, but I just wanted to know, what did the detergent, the tenderizer, and the alcohol do? The detergent and the tenderizer were used to separate DNA, from the cell. And the alcohol was used to float the DNA. Because again, the DNA does not dissolve into the alcohol. And then finally, number 5 here, what component is responsible for breaking down the proteins? Well, that is the soap mix. And then what term, it's soap mix and the tenderizer. And then what term is given to a molecule that specifically breaks down the proteins? Well, in soap, they put in chemicals, enzymes that are called surfactants. Surfactants are the materials that break down the organic material, so surfactants. And then there were some general questions that the student was to pick one and answer. Those questions included, and I'll go through each one and explain. From what other tissues can DNA be extracted? Basically anything that's alive on Earth has DNA material in it, and you can extract it. What different types of household enzymes would affect the DNA yield? Well, if you use salt, you might not get as much as with the specialized stuff in the meat tenderizer. If you were to use a cheapy soap instead of the more expensive stuff that we used, you might get less of a yield of DNA. What liquid laundry detergent additives might affect the DNA extraction? It could be the proteins and the phosphorus or the phosphates that they put in. How could you determine the yield? Are you able to weigh it in? Well, why not? Why not take your strawberry, put it on a balance, measure the mass, and then after you've got the DNA out, you could actually measure that DNA if you had a fine enough mass, and you could make a percentage of how much of the mass of the strawberry was the DNA. Would freezing a tissue first increase the DNA yield? This one brought the most discussion because most students don't realize that when water reaches about 34 degrees Fahrenheit or about 1 or so degrees Celsius, the water molecule expands. This is what causes ice to float. This is also what happens if you were to freeze these strawberries and let them thaw out. They immediately turn to mush, because when they freeze, the cell structure of every cell that contains water bursts. It's destroyed. If you were to freeze organic material before you took the DNA out, theoretically you should be able to get more DNA out simply because you have destroyed more cell structure and allowed that DNA to come out. And then, does the freshness factor of a tissue affect the DNA yield? Well, these strawberries have been in there now for weeks, and we actually did get much more DNA material out than just this. So I would hazard the guess that yes, the freshness of the DNA possibly has an effect, or the freshness of the strawberry does have an effect on how much DNA we can get out. And that, really quickly, is how you get DNA out of a strawberry. Hope you enjoyed it. And again, for those of you that have asked for lab papers from me. I apologize that many of them get lost when I move from school to school. I do try and keep them all in a thumb drive, and unfortunately many of them though do get lost because they're just tailored for that one instance in time. This one, however, this one's something that is relatively simple, materials that you should have around, most of you in your schools or even at home if you wanted to try it. And I will have a version of this one ready on Word if you'd like to ask for a copy. Thanks again. See you soon with another lab.